Jamurtas, and thank you for being here. Today's talk is going to be on oxidative stress and cardio-metabolic diseases. So the first part is going to be on what is oxidative stress, how is being developed, and the second part is going to be on oxidative stress and the cardiovascular disease development, and the final step is going to be on exercise, oxidative stress, and metabolism in general. So to begin with, we're going to start with a term of oxidative stress. What is oxidative stress? And I think most of you are familiar with this. When we talk about oxidative stress, we are talking about something that is referring to an imbalance between the systemic formation of reactive oxygen and nitrogen species, which is also known as RONS or ROS, and the biological system's ability to quench those reactive oxygen and nitrogen species. Those RONs are also known as free radicals, and from now on I'm going to referring to those as free radicals because it's a generic term and most of us are familiar with this one. We see here on the right side a balance, and on one side we see the ROS or free radicals, how they are developed, and there are three of those that are well known, and we're going to talk about those later on. And from the other side we see the second part of the balance, which is the antioxidant system, and we're going to talk about it later on. Let's see what's free radicals. We need to give a definition of free radical, and we're going to start by looking back at physics, and from physics we know that electrons are negatively charged and spinning pairs in orbitals around the nucleus. We know that nucleus is positively charged, so we have the electrons that run in pairs around the nucleus, and that is something that causes stability to the molecule. When an electron is going to leave this orbital, then a free radical is going to be formed. And we know that, as I said previously, that a stable molecule is the one that has a pair in the outer orbital of electrons. So when we lose one electron, a free radical is formed. When a free radical is formed, then can cause damage to several molecules in our body, like lipids, protein, and DNA. There are different causes, different ways of forming free radicals. Well, UV light, for example, if we stay in the sun for a long period of time, then the UV light is going to damage the skin cells, and that is when we're going to have a rash, and sometimes we're going to have a major burn. Ionizing radiation or smoking or air pollution are other ways of forming free radicals, and we have two more ways of forming free radicals. One is metabolism and the second one is inflammation, and we're going to look at those two later on. Since we're in an exercise and science department, I'm going to spend 2 minutes talking about how free radicals are formed during exercise, and we're going to talk, the latter part of my talk is going to be based on exercise, especially sedentary type of exercise and formation of free radicals. So I'm going to spend 2 minutes talking about how free radicals are formed during exercise. So one way is through leakage of electrons in the electron transport system. We know that electrons are moved, so at the end hydrogens are going to be form water with oxygen, and at the same time ATP synthase is going to form ATP, and we're going to have energy through this system. When this process is taking place, some electrons are leaving this normal process, and we said that when an electron is going to leave the outer orbital, then a free radical is going to be formed. So this is one way of forming free radicals during exercise, and especially aerobic type of exercise. A second one is through what is known in the literature as ischemia reperfusion, and we know that from the cardiologists who say that when they clamp an artery in the heart to perform the surgery, when they unclamp the artery, then some cells in the heart are going to be damaged due to the high presence of oxygen, and oxygen, as we said previously, is one of the ways that are going to form free radicals. So free radicals are going to damage part of the heart, and at the same time, the same thing can happen in our muscles and in general in other cells in our body. A third way is through neutrophils and oxidative burst. If we look here, we see different cells that are that we can see with blue, and here we see that something appears like an explosion has happened with red here, and this is what is called oxidative burst due to the high presence of oxygen here in the neutrophils that are going to destroy different viruses or different foreign things that are going to get into our body. And a fourth way during exercise that can develop free radicals is due to destruction of different molecules like hemoglobin or catecholamines that contain iron. And in the second part of my talk, I'm going to talk the relation between iron and oxidative stress, and this is a way of forming free radicals during exercise. So we see that during when we are talking about exercise, we have to separate what is happening during exercise and what is happening post-exercise. During exercise, we talked due to the fact that higher oxygen is going to be getting inside our body, free radicals are going to be formed, and post-exercise, the higher presence of free radicals are going to be due to the fact that some muscle damage has happened. So we're going to connect those things later on. Let's go and carry on talking about what is the role of free radicals in our body. Most of us connect free radicals with something bad, but the literature says that free radicals are good and bad. So here we are looking at the good role of free radicals. As I said previously, they can remove different debris. For example, when a muscle is damaged, we need to remove those proteins that are present in the muscle, and this is happening due to this mechanism here, due to oxidative burst. And secondly, we have fertilizations. We know from the literature again that when low levels of oxidative stress is present, then fertilizations can be greater. And thirdly, the immune system. We know, for example, that in order to get rid of different viruses in our body, we use oxidative stress to destroy those. So the bad role, though, of free radicals is that it can damage biomolecules, it can lead to diseases, as we're going to talk about cardiovascular diseases later on. And finally, there is a theory of aging that connects free radicals with the destruction of our body, that at the end of the day, we're going to eventually die due to this fact, meaning higher formation of free radicals. In order to quench those free radicals, as I said previously, there is a system that is called antioxidant system. And this is a mechanism to quench free radicals, and it has two parts, the enzymatic and the non-enzymatic. And what does an antioxidant do? It can donate an electron, and we see here a free radical. By definition, this molecule does not have a paired electron in the outer orbital, and we see that this antioxidant is going to come here and donate an electron, and this molecule is going to get stabilized. A different problem is going to be developed, though, since this antioxidant is going to have an unpaired electron, and I'm going to talk how this can be blocked later on. So we have different enzymes in our body, antioxidant enzymes. I present here 3 of those: the superoxide dismutase, catalase, and glutathione peroxidase. So in our lab we use some of those enzymes, we measure some of those enzymes to see whether oxidative stress is present. And secondly, there is the non-enzymatic antioxidants, and there are different vitamins, there are different coenzyme Q10, for example, ferritin, albumin, bilirubin, ceruloplasmin, flavonoids, uric acid, and thiols. And I present here vitamin C, vitamin E, C, and A, which are thought to be antioxidant vitamins, and I'm going to talk for vitamin E and vitamin C later on. And here we see 2 other molecules, uric acid, which is believed that 50% of the antioxidant capacity of the plasma is due to uric acid, and there is a specific metabolic pathway that is going to lead to uric acid, and we can explain why uric acid is presenting with such a high antioxidant capacity, but this is beyond today's talk. And finally, we have glutathione that is believed to be one of the strongest antioxidants in our body, and is present in very large levels, and it can destroy free radicals. The synergistic action between, as I said here, vitamin E and vitamin C and glutathione, we can see it here. When a radical is formed, and in the literature a free radical is presented in short with a capital R and a dot, meaning that is a radical with an unpaired electron. Vitamin E is going to donate an electron, and the radical here is going to come back to the reduced state, whereas vitamin E is going to be present here in oxidized state. Oxidized vitamin E is going to come back to the reduced state through the action of vitamin C, and vitamin C, now that is oxidized, is going to come back to the reduced state through the action of glutathione. So we see a synergistic action between different vitamins that are part of the non-enzymatic antioxidant system with the enzymatic system, which is present here with the glutathione peroxidase and the glutathione. So that what's happening in our body in general, a synergistic action of different molecules. Free radicals are not staying in our body for long, and we can understand that through the half-life of these different free radicals. We see, for example, superoxide, hydroxyl radical, nitric oxide, the hydrogen peroxide. Those are the half-lives, and we can see, for example, for superoxide, the half-life of this molecule is 10 to minus 6 seconds. See here, seconds. So it's impossible to measure those molecules. So what do? We usually use indirect indices to measure either lipid peroxidation or protein carboxylation, and we infer that higher presence of these molecules are going to be due to higher presence of oxidative stress. For example, we have exhaled pentane, MDA or T-bars, lipid hydroperoxide, isoprostanes, and conjugated dienes that are part of assessing lipid peroxidation. We have protein carbonyls to see whether proteins have been damaged. We have the glutathione either in the reduced or the oxidized state. We have the total antioxidant capacity of plasma, uric acid, bilirubin, electron spin resonance, vitamin C, and vitamin E. There are 15 of those indirect indices that we can use to measure oxidative stress, and with red here are the ones that we are usually measuring in our lab, because I believe that you cannot use just one index to measure oxidative stress. You need to have a cohort of indices in order to get a better picture of what's going on with this system. Let's move to the second part of my talk now, which relates oxidative stress and cardiovascular disease. And I'm going to talk about atherosclerosis, and atherosclerosis is a process in which an artery is blocked due to the formation of a plaque or thrombus, and if the thrombus is formed in the coronary arteries and the blockage is significant, then a heart attack can take place. We see here the formation of a plaque. Then if this is significant and the blood flow is going to be reduced, then a chest pain can happen. Atherosclerosis is formed through the formation of those cells that are called foam cells, and those foam cells are happening due to the oxidation of LDL. And LDL can be oxidized. LDL, we all know that LDL is the bad cholesterol, but it's not only the LDL that is going to significantly contribute towards atherosclerosis formation due to the formation of foam cells, but it's the oxidized form of LDL. And LDL can be oxidized through different ways. I present here 4 of those: smoking, diabetes, hypertension, and the high levels of LDL itself is a significant factor towards LDL oxidation. Here on the right part of this part, we see here an artery that is unblocked, a clear artery. We see the lumen of the artery, how good it is, and blood flow can get through here. Now, there are 2 different types of cells in our arteries that are going to significantly change its formation, and they are going to form foam cells. Those are the smooth muscle cells and the macrophages, and the dietary fat, high dietary fat, is going to at the end increase the levels of LDL, and if LDL is oxidized, then, as I said previously, that is going to cause the formation of foam cells in the smooth muscle cells and macrophages. How does this process happen? And we said that the connection between the cardiovascular diseases and oxidative stress is due to the fact that LDL is going to be oxidized. How the foam cells are going to be formed? For example, we see here the endothelium. This is the intima of the artery. This is the endothelium. When something is going to happen in the endothelium, then platelets are going to get over there, fibrin and monocytes are going to get over there in order to clear the damage that has happened in here. Adhesion molecules are going to develop in here, and we see that monocytes are going to get to this particular part of the endothelium, and at the same time we see that cholesterol and lipids are getting over there. Once this happens, then monocytes are going to get through the endothelium inside the intima, and we see that monocytes are going to develop, are going to change, are going to be macrophages, and we see here those yellow things are the LDL that is oxidized. It's going to get inside macrophages, and this is a foam cell that has been formed. At the same time, we see that smooth muscle cells down here are going to move inside the edema and are going to get the oxidized LDL and are going to turn into foam cells. And what is happening? Those foam cells are going to move, are going to push the endothelium towards the lumen of the artery, and at the end is going to decrease the lumen, and blood flow is not going to get to the same extent as has happened before. And we know that the human body is very adaptable. We see here the popliteal artery that is coming down the legs, and right here popliteal artery is blocked. So that means that we are not going to be able to get, if this happens at once, that means that blood cannot go down to the leg. So what is what's the adaptability of our body? What has happened to our body in order to get rid of this problem? We see here the collateral vessel formation that is going to get blood through this point, and it's going to come down, the blood is going to get down to the leg. So, of course, we're going to have problems. It's not the same thing, but at the same time, but at the end, I'm not going to have, you know, my leg chopped off due to the fact that the popliteal artery was blocked. Now, let's see why oxidative stress is important in the LDL formation and in atherosclerosis in general. We say here that iron, oxidative stress, and cardiovascular diseases are interconnected, and we're going to see how. We know from the literature in chemistry specifically that there are different reactions that are called Fenton reactions that connect iron with oxygen, and we're going to have a free radical formed. Same thing happens with copper due to the Haber-Weiss formation, and at the end of the day we see that LDL is going to be oxidized. We see here we have a hydroxyl group here that with LDL is going to oxidize the LDL. And we said previously that oxidized LDL is the molecule that is going to turn into foam cells when it's going to get into the smooth muscle cells and monocytes, macrophages in particular. How do we know that? How we can interconnect reactive oxygen and nitrogen species with iron and atherosclerosis? The first thing that connects those three is a condition that is called hemochromatosis. It's a genetic abnormality that these individuals tend to store high amounts of iron in their body. And we see here that as the body iron content in grams is increases, we see that the different steps of this disease, that at the end of the day, we see that before they reach the age of 50, these individuals are going to die. Some say that the mean age of death for these individuals is in between 20 and 30 years of age. Why is this? It's because high levels of iron are going to lead to the formation of free radicals. Free radicals are going to change LDL into oxidized LDL, and LDL is going to form foam cells. Cardiovascular diseases are going to develop. So, how do we know, again, how reactive oxygen and nitrogen species are related to atherosclerosis? We see here the therapy of this particular condition. The therapy is through, in the beginning, is through blood donation. Why is this? Because one vial of donated blood accounts for 250 milligrams of iron. And we do that once or twice a week. So what do we want to achieve here? We want to get rid of iron, because iron through the Fenton reactions are going to lead to the oxidized LDL, and at the end of the day is going to cause cardiovascular disease. We monitor ferritin and hemoglobin levels because we want to see what's going on with iron levels. Okay. Again, we know that we can connect iron, oxidative stress, and cardiovascular diseases through the fact that there are reports in the literature that indicate that when small children take accidentally iron tablets, they can die acutely due to the fact that their cardiovascular system is going to be damaged. Furthermore, we know that there is way back, like 25 years ago, there was a doctor in the United States that said that it's not the cholesterol that is the problem with the cardiovascular diseases. Because there is someone that can have high levels of cholesterol, but if that cholesterol is not going to get into the oxidized form, LDL is not going to get into the oxidized form, then foam cells are not going to be developed. So he believes that iron is the problem, because we see here, this is a ferritin, here is oxidized LDL, and this is the odds ratio for carotid plaque. When I say carotid plaque, I mean the formation of thrombus. And we see as the levels of oxidized LDL increase, and at the same time the ferritin levels, which show us what the iron levels are in our body. So we have two things here. One is high levels of iron, high levels of oxidized LDL, the odd ratio for increased carotid plaque, and in general development of a cardiovascular disease is increased. So he believes that LDL is the gun, oxidized LDL is the gun, but the iron is the problem that is going to pull the trigger, and foam cells are going to be developed, and atherosclerosis is going to be present in our arteries, and at the end the cardiovascular disease is going to be developed. Lastly, I'm going to talk a bit about oxidative stress and metabolic diseases, and in particular I'm going to focus my talk on the eccentric type of contractions because lately if you hit on PubMed eccentric exercise, you're going to get more than 10,000 hits back. So eccentric exercise, unaccustomed eccentric exercise can lead to increased muscle damage. Muscle damage is going to lead to increased inflammation. Inflammation is related to increased reactive oxygen and nitrogen species, as I said previously, that are going to have two things. They are going to cause two things. One thing is going to repair the muscle tissue, because I said previously that when I have a muscle damage, and proteins are going to change its structure, then I need to get rid of those. Think of a house that an earthquake has hit the house, and the house has been destroyed. If I want to rebuild the house, I need to get rid of the debris. So who does that? Reactive oxygen and nitrogen species are going to do that. How do they do that? Remember what I said about oxidative burst. If we go back, way back, when we talked about the different ways of forming free radicals during exercise, one of those things that we said is neutrophils and oxidative burst can lead to the formation of free radicals, and one of the things that are going to happen is we're going to get rid of those damaged proteins. So one thing that is happening is we get those reactive oxygen and nitrogen species, free radicals, are going to repair muscle tissues. And the second thing they're going to do, they are going to upregulate the antioxidant activity of the particular muscle, and these muscles are going to be resistant to muscle damage when the same protocol, exercise protocol, is going to be produced again by those individuals. So this thing here is known in the exercise literature as the repeated bout effect. So the repeated bout effect and oxidative stress, we ran a project a few years ago, and we have here we took subjects inside the lab, and they performed 5 sets of 15 maximum eccentric repetitions. In total 75 maximum eccentric repetitions, which is stressful to the muscle, and muscle damage was developed. So we wanted to see, this was run, as I said, a few years ago, whether muscle damage is going to be related to oxidative stress. So we see that oxidative stress 2, 3, and 4 days after the exercise, when muscle damage is present, remember that delayed onset of muscle damage appears not at the end of the exercise, the unaccustomed exercise that I have performed, but is going to be delayed, and usually peaks 2 to 3 days after the end of the exercise. So we see here 2, 3, and 4 days that the oxidative stress, the redox status is perturbated. We see glutathione, catalase, catalase, protein carbonyls. For example, glutathione 2, 3, and 4 days is reduced due to the fact that glutathione was utilized in order to get rid of those proteins, damaged proteins. And when we performed 2 weeks later the same protocol, oxidative stress was less. So less muscle damage, less oxidative stress. So an adaptability has happened to the muscle tissue, and at the same time less oxidative stress was developed. How is this related? How is oxidative stress is related to metabolism? If we look at lipids now, and we know that lipids are associated with atherosclerosis, triglycerides, total cholesterol, HDL, and LDL, same protocol. For example, triglycerides 2, 3, and 4 days following the end of the exercise session was lower. And if I do an association with the muscle damage being present and oxidative stress, I'm going to see a good correlation. So when muscle damage is present, we're going to have less when less muscle damage is going to be present, I'm going to have less oxidative stress, and I'm going to have less lipids. Why is this? Because those lipids, for example, cholesterol is going to get to the damaged tissue, and it's going to be utilized to form new cells. Remember that when I have a cell membrane is composed of cholesterol. So I have in my blood less cholesterol because it's going to be utilized to form new cells, new cell membranes. And if I look, we run another experiment and we see that the perturbation in the lipid profile is different between overweight women and normal weight women. So it has to do with body type and in general how the BMI as well. Another thing that you have to keep in mind, because here we're talking about acute exercise, there were these individuals here from Austria that ran a very clever experiment to associate eccentric exercise that is going to cause muscle damage, but at the same time is a different type of exercise with metabolism. Training here, we have 8 weeks of training, and they took individuals to Pertuli, a similar Pertuli, which is a ski resort, and they took individuals with the lift uphill, and they had them come down on foot. When they come down on foot, they perform eccentric type of contractions, correct? Because downhill is going to elongate the muscles, and they did only eccentric type of contractions. They had a different group that they were going uphill on foot, but they were coming down with a lift. So two different type of contractions. Going down eccentric type, going up concentric type of contraction. If we look here at the lipids, we see that LDL cholesterol and ApoB, ApoB is one apoprotein that is related to atherosclerosis, both concentric and eccentric type of exercise reduced their levels, reduced LDL cholesterol and reduced ApoB to ApoA1 ratio, which is an index of how prone are towards atherosclerosis. And we see here that eccentric type of contractions, which is one of type of contractions that we are utilizing in our lab, decreased LDL cholesterol and ApoB. At the same time, insulin levels, C-peptide and HOMA, which is an index of insulin resistance, were decreased due to these type of contractions. So if we are going to conclude this talk, we can say that oxidative stress can lead to atherosclerosis due to the formation of foam cells. Excessive iron intake can lead to oxidative stress and atherosclerosis because iron can change the formation of LDL. It can oxidize LDL. Unaccustomed exercise leads to muscle damage, and muscle damage is associated with increased inflammation and changes in redox balance. And the aforementioned changes are associated with changes in the metabolic profile, and chronic eccentric training is associated with positive changes in the metabolic profile. So with that, I thank you a lot, and questions are welcome.